right, good morning, good afternoon, and good evening, everybody. It's my great pleasure to launch the Cancer Center seminar series here at the Sanford Burnham, La Jolla, and I'm grateful to have you join us from Europe, Israel, and all the United States. We start a new series that actually is organized by Swasti here in our institute, Swasti Haricharan, and she will come on later on and she will moderate the questions as we go through the sessions today. The Focus On series is unique as we have two speakers in 1 hour, more or less, focused on one topic. And we have chosen to have a rising star as well as an established scientist in the field. And today we are fortunate to have Ravid and Tom as the two experts that lead the discussion on the tumor microbiome. I will start by introducing Ravid. Ravid Straussman is a scientist at the Weizmann Institute in Israel. Ravid got his bachelor degree summa cum laude at the Hebrew University, Hadassah Medical School. He earned his master degree in medical biochemistry and then did his MD Ph.D. also in the Hebrew University in Jerusalem. He did his internship in one of the biggest hospitals in the Tel Aviv area, Beilinson, and then did a PhD and postdoc, started in the Hebrew University, medical biochemistry and human genetics, and later on he was at the Broad Institute, MIT, with Todd Golub, where he further continued his postdoc training. Before he came back to the Weizmann, where he established his laboratory a few years ago. He's renowned for his discoveries of the microbiome in tumors, discoveries that changed our understanding of the field. And I'm thankful for you, Ravid, having the opportunity to share with us your recent studies and your presentation. All yours.
Okay, great. So thank you very, very much for inviting me today. It's a pleasure, and I'll try to tell you briefly in 25 minutes about things we did and we learned about the tumor microbiome, where we are and where we're going in this field. So my lab in the last few years have been interested in understanding the complexity of the tumor, understanding how different components of the tumor microenvironment affect the sensitivity of cancer cells to drugs. And we actually stumbled just by chance on studying the tumor microbiome. So when I was a postdoc in Todd Golub's lab, I looked at the way that stromal cells affect the sensitivity of cancer cells for drug. We did high throughput screens and found hundreds of cases in which merely by adding stromal cells to cancer cells, it completely shifted the sensitivity of the cancer cells to drugs. And one of these cases was the case of gemcitabine. So when we added gemcitabine to different cell line, and this is a case of RKO, colorectal cancer cell line, we found that it kills the cells. But if we added this specific stromal cell line called HDF, or the preconditioning media of this cell line, it completely rescued the phenotype, and the cancer cells could continue to proliferate under chemotherapy. So we were very interested to try and understand the mechanism of that. We looked a lot, but eventually, and making it really short, we found that our HDF cells were contaminated with mycoplasma. And none of the other cell lines, stromal cells that we used, had mycoplasma. This is a positive control and negative control. And we were able to show that it was a mycoplasma rather than the stromal cells that had the effect. And we also were able to figure out the mechanism. I'll show you this in a second. I'll just say first that to understand if this is something that is relevant to human biology, my PhD student, Lior and Michal, they explored the pancreatic cancer, and they asked, can we find bacteria in pancreatic cancer? And in different ways, we're able to show that the majority of pancreatic cancers have bacteria in them. This is by qPCR. but we also been able to detect them by staining. Here we're looking at LPS. We detected the 16S of the bacteria by fish staining and other methods. And again, to make a long story short, we found that the mechanism by which the bacteria protected the cancer cells from chemotherapy was degradation of the drug. So usually when gemcitabine gets into the cell, just like cytidine, it's analog, it's being phosphorylated 3 times, incorporates into the DNA. But what we found is that if you have bacteria inside the cancer cells, the bacteria pump in the gemcitabine very, very efficiently using the NAPC transporter, and then they degrade, they inactivate the drug, and that's how they protect the cancer cells from gemcitabine. So it's not that the bacteria really secrete something that rescues the cells, but rather they degrade the drug and protect the cells. And by a series of knockouts in bacteria, we're able to find a specific enzyme, which is what we call the long form of CDD. So the cytidine deaminase gene, what we found is that some bacteria, or 44% of all bacteria, do not have the CDD gene at all. Some bacteria, another 44%, have what we call the short form of CDD, which make tetramers. But we found that these tetramers, they cannot degrade gemcitabine. And only the 11.4% of the bacteria in the bacterial kingdom, we surveyed almost 3,000 of them, that have the long form of CDD, they make these dimers. And these dimers can efficiently degrade gemcitabine. When we asked which bacteria have the long form of CDD, we found that almost entirely they belong to the gamma proteobacteria class. So we then asked, so are these really, can these bacteria be found in human pancreatic cancer? And we sequenced the bacteria in 65 additional patients and found that indeed the most prevalent bacteria in these patients, what you see here in blue, come from the gamma proteobacteria class. And we also were able to demonstrate, I'm not showing here today, that if you isolate these bacteria and grow them, they can indeed degrade gemcitabine. And we showed in mice experiments that different models, I'm showing here one example, that if we have here this example of the CT26 colorectal cancer cell line that we put in immunocompetent mouse, we inject bacteria IV at home to the tumors, and we can show that if there's no bacteria, the treatment with gemcitabine can control the growth of this tumor. But if there are bacteria, the tumor is resistant to chemotherapy, as one would expect from the presence of bacteria that can degrade the drug inside the tumor. One of the things we didn't know back then is it really relevant to human? So we did find bacteria that can degrade gemcitabine, but are there enough bacteria that they can actually make a difference, a clinically relevant difference in the way patients respond to chemotherapy? And we didn't know if treatment of these bacteria would actually change the course of response. So I just want to point out a few studies that came out in the last year or two. In this very nice Cell paper by Florence and McAllister from MD Anderson, they followed a cohort of pancreatic cancer that had a short-term survival and a long-term survival, and tried to understand what the difference between these cohorts. And one of the differences, or the main one, was they found different bacteria that were present in these two different cohorts. And interestingly, when these bacteria were used, or bacteria from short and long-term survivors were used as FMT for mice with a KPC pancreatic model, they found that mice that got the bacteria FMT of bacteria from short-term survival had a faster, more rapid growth of the tumor, but mice that got bacteria from the long-term survivors had a slower growth of tumor as compared to the controls. And the mechanism behind it also probably involved effect on the intratumoral immune response, as can be shown here, that the mice that got the bacteria from long-term responders, they had higher amounts of CD8 positive lymphocytes in the tumors. But in the last year, there came 2 additional papers that, following our work, looked retrospectively at patients with pancreatic cancer. And they asked, they said probably some of these patients got some antibiotic treatment even just by chance. And both works seems to support the same outcome, that patients that got antibiotics, specifically quinolones like ciprofloxacin, as we showed in our paper, they actually survived better. The overall survival was much better for patients that got quinolones versus patients that were not treated with quinolones. And we are, I must say, planning now a prospective study in order to show similar results. So one of the things we did over the last few years was a lot of high throughput screening, looking again at many cancer cells and drugs, but trying to understand how different bacteria can change the way cancer cells respond to therapy. And I'm not going to show many of these results right now, just say that we see many, many hits. So we see bacteria that can shift the response to a large number of cytotoxic drugs, targeted therapies, and also immunotherapies. And we're following many of these hits. Some of these I'll show later on. Now, as you know, bacteria have been known to be present in tumors for many years, probably over 100 years. But I think these 2 seminal papers looking at the presence of Fusobacterium nucleatum in colorectal cancer really made a shift in this field. And from these papers and on, there have been more and more papers demonstrating the presence of different bacteria in tumors, and also exploring very nicely the effect that these bacteria have on tumor biology. And we assembled a team in the lab led mostly by Dvora and Ilana and Gary and Noam Shintal from the Open University. And the idea was really to try and characterize in an unbiased way a large number of tumors and see which bacteria are there, where are they, and many other questions related to the tumor microbiome. Last year, we published this study, and overall, we had more than 1,500 samples. We had a very large number of negative controls because one should be very, very cautious when exploring the tumor microbiome because of the low biomass that is there, not to confuse contaminations with real data. We had samples from 9 medical centers, which we found to be critical, as we found a lot of center-specific contaminations. We optimized our DNA extraction with a novel bacterial 16S PCR method in which we amplify 5 regions, which give us a better resolution. And we did extensive visualization. So what did we find in this study? So overall, we found that the bacteria are present in all of the tumor types that we looked at. This was melanoma, lung cancer, ovarian, GBM, pancreas, bone, and breast, and also colorectal, that is not on this graph. But more than that, we found that different tumor types seems to have different bacteria. So here you can see it looking at the phylum level. As expected, and then as we showed in our previous study in pancreatic cancer, one can mostly find the proteobacteria. And as you can see here in colon cancer, as expected, we can find mostly firmicutes and Bacteroidetes. And if you look at specific species, it seems that they cluster nicely based on the tumor type. So one can find that specific species are enriched in specific tumor types. And indeed, when you look at the Jacquard similarity index between all possible pairs, it seems that tumors are always more similar. Tumors of the same type are always more similar than the bacteria in these tumors to bacteria in other tumor types. And I should mention also that in a really beautiful paper that came from Rob Knight's lab, Greg Poor showed earlier last year, looking at TCGA data and extracting only the non-human reads, that they can also find bacteria across many different tumor types, but more than that, that the bacteria are so tumor type specific that they can actually predict the tumor type in many cases, just based on looking at which bacteria are found in these tumors. We also found that Fusobacterium specifically, because it's a very interesting bacteria, is present not only in colorectal cancer, but can also be found in breast cancer, pancreatic cancer, and other cancer types. As you know, there have been a lot of literature in the last few years demonstrating that Fusobacterium has impact on cancer, both in the transformation process, affecting proliferation, suppressing the immune system, promoting chemoresistance. So we thought it's very interesting. And together with Gilad Bachrach, we collaborated to demonstrate that just like as in colorectal cancer, we can see upregulation of galgal-NAC. This is the ligand for which Fusobacterium home through its FAP2 receptor. So we see that there upregulation of galgal-NAC in breast cancer as compared to a normal breast. And more than that, they demonstrated that when you inject Fusobacterium nucleatum into mice model of breast tumor, you see that the faster growth, higher tumor volumes of Fusobacterium. If you inject a FAP2 deficient Fusobacterium, which cannot home to the tumor, you don't see this effect anymore. This effect is probably mediated by the suppression of TILs like CD8 and CD4. This was published last year. We also compared some bacteria in tumors versus normal, demonstrating that in breast cancer specifically, we can find many more bacteria in the tumor tissue as compared to the normal adjacent tumor and as compared to a completely normal breast, as you can see in these rare fraction plots. We were also very interested to find the localization of these bacteria. We stained hundreds of tumors in different methods. And I think the bottom line is that we mostly find the bacteria in the cancer cells rather than in stromal cells and also in immune cells. Here you can see an anti-LPS staining of human breast cancer. And you can see that the LPS staining is confined mostly to the epithelial compartment. This is an electron microscopy demonstrating bacteria in the cytoplasm. We frequently notice that the bacteria lie very close to the nuclear membrane envelope. This is not by chance. Here we can see additional, this is a CLEM in which we do both TEM and immunofluorescence. So you can see the bacteria, anti-LPS here, and you can actually see the bacteria in electron microscopy, again, very closely related to the nuclear envelope, which you can see here. These are bacteria that we made GFP positive, and when we infect them into cancer cells, we see again that they're going to the same perinuclear location, which we don't fully understand why. And we can also see bacteria, say, in many immune cells. These are inflamed breast cancer from a patient. You see there's many CD45 cells by immunofluorescence, and same cells seems to be positive, in this case, by FISH to bacterial 16S. We also see many times macrophages in tumor that are loaded with bacteria. Here we stain them for LPS, as you can see here, and LTA and CD68 to show that these are really macrophages that are loaded with bacteria or bacterial remnants. So if there are bacteria inside cancer cells and inside immune cells in the tumor, can you expect some of their antigens to be presented on the MHC class 1 and 2 of tumor cells? And I'm going to very briefly show a collaborative work that we did. It was led mostly by Yardena Samuels, with collaboration also with Jen Wargo and students from Yardena's lab and my lab. And what we did here, we took melanoma samples from 9 patients, so overall 17 metastases. And we identified both the intratumor bacteria in these, but also HLA peptidomics. And we specifically asked, can we find bacterial peptides that are presented either on MHC class 1 or 2 on the tumor and immune cells? And the answer was yes. We could find overall peptides of 41 bacteria in these patients, identified across these patients, as you can see here, many different bacteria. And we also validated it by loading the specific peptides that we found, in this case, in this patient from Fusobacterium. We loaded these peptides on EBV transformed B cells and then co-culture it with autologous TILs from these tumors to demonstrate that there's a subpopulation of TILs that actually recognize specifically these bacterial peptides. So we think this is also, it gives some other ideas of maybe more treatment-related ideas, but at least I think it also shows that not only that the bacteria are present in the cancer cells, they can also, some of the neoantigens are probably presented by these cells. So are there live bacteria inside human tumors, or are we just staining tumors that are not alive? We had a big campaign of biopsy from patients, mostly breast cancer patients. And in many of the cases, we can grow bacteria on 70 different types of agars that we have in the lab, both in aerobic and anaerobic conditions. And we're now in the process to sequence almost 1,000 of these different bacteria that we isolated and studying their genomes, trying to find out their adaptation to the tumor microbiome. We can also take biopsies from patients, as you can see here, from a breast cancer patients, and we can grow them ex vivo. And then we can label them with D-alanine that is specifically incorporated only to the peptidoglycan of bacteria. And again, we can see specific cells in the human tumors that are loaded with bacteria and become positive for D-alanine staining. So why do we see this tumor-specific bacteria preference? We think that it has to do with the different functions that these bacteria carry with them. And I'll give you a few examples. Here we looked at the metacyte functions of these bacteria using PyCrust-2. And as you can see again, we see some functions that seems to be specific to each of the different tumor types. And just to give you two examples, we see that bacteria that come from bone, they are enriched with enzymes that can degrade hydroxyprolines. Then you can, as you probably know, hydroxyprolines are a product of bone degradation and that are highly enriched in bone. Or another example, if one compare the microbiome of lung cancer between never smokers and current smokers, we find some bacteria that are enriched in the smokers. But when we look at the functions, it makes a lot of sense. We see that the functions are degradation of nicotine, or toluene, or acrylonitrile, or chlorobenzene. These are all chemicals that are found inside a cigarette smoke. We also compared the presence of these bacteria in tumor progression and response to anti-cancer therapy. And again, just to show you snapshots of some of the results, we found many interesting correlations. Here, for example, we found different amount of bacteria between breast cancer patients with an early recurrence or late recurrence. We also, as you know, there have been really beautiful works also from Tom and others looking at the effect of the gut microbiome, how it modulates the response to adding PD-1. And just recently, there were two nice papers showing that by modulating the gut microbiome by FMT, one can resensitize melanoma patients that were refractory to anti-PD-1 therapy to immune checkpoint inhibition therapy. And we found that if you compare tumors from melanoma patients in this case that responded or did not respond to immunotherapy, you can find interesting bacteria and also interesting functions that are enriched in the responders or non-responders. So as a summary of this short presentation, as I told you, we found that bacteria are frequently found in many types of human solid tumors. I think there's really a lot of data now from many, many groups around the globe demonstrating the same observation that intratumor bacteria are present mostly, we think, in the cancer and the immune cells. Different tumor types have different composition of intratumor bacteria. I didn't show you, but we found that breast cancers has a richer and more diverse microbiome than other tumor types. We think that intratumoral bacteria may affect tumor growth and the response to treatment, and we're following many of these projects. And right now, the main studies that we do in the lab in the microbiome field is we have, as I told you, we have a few projects looking at the effect of bacteria on drug resistance, including immune checkpoint inhibitors. For example, we just concluded a large clinical trial in which we followed hundreds of patients that started to get anti-PD-1 therapy. We followed them clinically, but also characterized their tumor microbiome, and we found a very interesting correlation there. We're very interested in following the cancer and bacteria crosstalk for this many times intracellular bacteria. We're interested to understand more about the homing of bacteria to the tumors and the invasion mechanism to the tumor and the immune cells. We also have a few projects in which we try to manipulate the tumor microbiome in different ways and try to see how it optimizes the response to therapy. And also a very large project that we've been conducting for many years, looking again at hundreds and hundreds of tumors and profiling the tumor microbiome, and also the correlations between the microbiome that we do find in some of the tumors and the microbiome in these tumors. And just very important for me to say, of course, thank you for the very, very many dear collaborators that helped us along the years, specifically Dan Raz and Jen Wargo, Daniel Pieper, Kristen Blank, Keith Flaherty, Nicola Balladini, and Sophia Wendt, and many more collaborators. And of course, I need to thank my group here at the Weizmann Institute and many other collaborators and our funding agencies that let us do this work. And thank you for your attention.
Wow, thank you so much, Dr. Straussman. That was an amazing talk to kick off this seminar series. We have quite a few questions coming in, but I'm going to take the privilege of being the moderator to ask you the first one. I'm really curious as to why you think the tumor cells, or even like the pre-neoplastic progression areas, have more bacteria than the normal cells. Is it because the tumor itself is producing some sort of immunosuppressive environment that's allowing the bacteria to grow, or is it the other way around, that the bacteria are creating an immunosuppressive environment that's allowing them to grow within the tumor cells?
Yeah, so my guess it will be that there is a mix of both. I mean, some bacteria are known to be transformative. Even Helicobacter pylori, for example, in gastric cancer, right, has a transformative effect. But some bacteria might be hitchhikers, that they get to tumors just because these tumor has immunosuppressed environment, sometimes anaerobic environment that is good for them. You know, the different metabolites that they find in these tumors. Also, the access to these tumors is easier than to normal tissue, as the neovascularization of these tumors. Many of these blood vessels are fenestrated to some way. So I think there's many reasons why bacteria would specifically prefer to grow in tumor rather than in normal tissue.
Totally fascinating. And someone had a related question on Urban Roy Chowdhury, and they were wondering, do the low number of bacteria in the tumor microenvironment indicate a fight or a competition between proliferating tumor cells and growing bacteria for the limiting nutrients? So can that also explain the specificity of the species you're seeing that gets selected in a particular tumor microenvironment?
Yeah. Much like this is a puzzle for us to some extent. How can there be a bacteria there, but we don't see we don't see a sepsis, right? We don't see an active bacterial disease, and still some of the bacteria that we find sometimes are pathogenic, not always. So we're not really sure what keeps these bacteria in check. It's very hard for us if we try to grow bacteria in culture with cancer cells. Usually they proliferate fast, and they kill the cancer cells. And it's hard for us to culture them long time side by side together. So there's something there that keep everything in balance. Probably the immune mechanism is involved, but we don't have enough to understand this.
Yeah, related to that, Alexander Arzamasov asks, what is the origin of these bacterial cells in the different tumors? How do they get there? Is it just they are already present in the normal tissue?
So when we compare the microbiome of a tumor and normal, we definitely see some similarity that is not by chance. So we do think that some of the bacteria in the tumors originate from the normal tissue when there is a microbiome in the normal tissue. Some other get to the bloodstream. I think there were a few really nice papers in the last 2 years, for example, demonstrating colorectal cancer, that the fusobacteria that is frequently studied there is not coming from the gut lumen, but many times probably from the gingiva through the bloodstream to the tumor. So I think some bacteria probably get there. We showed a few years ago that some of the bacteria that get to pancreatic cancer probably get there in a retrograde manner from the duodenum back to the pancreatic cancer. So there's probably many different answers to this question.
Wow. So there's a bunch of sort of related questions getting into what you think the mechanism might be, whether it's the C-gas sting pathway with the association between these microbiota and tumor progression and survival outcomes, or is it because of the recruitment of tumor immune cells? What do you think is the most likely mechanism that's causing this this phenotype? Could it be that the bacteria are actually a vehicle for gene transfer?
I don't know much about this. I mean, there have been, I think, a lot of literature demonstrating interesting crosstalk between bacteria in cancer cells, and the effect can be indeed immune mediated. There have been people show the effect on the Wnt pathway, and of course in STING and kappa B. And I think there's really a lot of crosstalk between bacteria and cancer cells. So it's not, I would guess, not one dominant mechanism, but different bacteria, different cancers, you would have different mechanisms involved.
So I'm gonna,
Ravid, am I allowed to throw a question in there? Because one thing that I've wondered is sort of related to one of the posted questions, which is, do you think the ability of a tumor cell to allow bacteria in is a marker for loss of an innate immune signaling pathway that otherwise would have countered the bacterial presence? So I think that's one of the origins of the STING question. We know some tumor cells retain STING pathway activation as one example, but many lose it by one mechanism or another. So I'm curious, I mean, it seems, unusual for a cell to allow bacteria in because there are strategies to keep bacteria out or to kill them. So maybe it's a marker for loss of a key pathway in the tumor cells. What are your thoughts about that?
Yeah, I think you're exactly on point. You got it right. And we're actively asking what these changes are. And the one thing we're doing now, we're trying to do a high throughput screen in which we in vitro test bacterial invasion into many different cell lines. And then we're comparing the genetic changes in these cell lines to the success of cells, not only to invade, but also to survive in these cells. We have some preliminary data linking specific genes, but this is not established enough, but we're on it. I think this is exactly where we're going.
We have a couple of minutes left to ask questions, so I'm just going to sneak in some quick ones. I think this one's really interesting from Ben Lewis. What do you think your work means for how we should plan, say, mouse experiments for our tumor xenograft studies? Should we be giving these mouse models of cancer? Should we be treating the mice with antibiotics to see if there's an effect of bacteria in the microenvironment? What should we be changing about how we plan our experiments?
Yeah, so I should say that it really depends on the model. So in my xenograft that everyone very commonly use, we usually don't see bacteria. These models, they grow very fast. I know in my lab, in others, I think you inject cancer cell lines into, especially if it's a syngeneic mouse model or even a nude mice, and in 10 days, in 2 weeks, you have an established tumor. And usually we find 0 bacteria in these tumors. So I don't think one should change anything. When we go to genetically engineered mice models, then we start seeing bacteria that natively get into the tumor. These tumors, as you know, many times grow. It takes them months to develop. And sometimes these bacteria also reflect to some extent the bacteria that we see in the human related tumors. So these type of tumors, also immunocompetent tumors, can be a great field for experiments in looking how modulating the tumor microbiome change different phenotypes of the tumor. These are just very long experiments that take a long time to execute.
Okay, let's do a last question, and then we'll come back and revisit some of the questions we couldn't get to in the last 10 minutes after Dr. Gajewski gives his talk. But Bohu asked this question, showing that your fish images seem to show more bacteria and tumor cells than in prior studies. Do you have any comments on that? Is it a different probe, or is
our fish experiments?
Yeah.
Yeah, so we were very worried about, I must say, off-target effects, and I'm still worried about it. So we did every possible control we could think of. This includes, you know, a random sequence, shuffling of the sequence of the fish, which was completely clear. And still, I must say that I also feel the same, that sometimes it seems to be a lot of bacteria there. And I was surprised by that. The one thing I could do is just report on our results. But if someone come in a year or two and say, you know, we found some off-target effect that you didn't think of, I'll be more than happy. We're also trying to control for this all the time. We try to do many different modalities, looking at immunohistochemistry with antibodies, looking at fish, which is completely different, looking at electron microscopy, culturing these bacteria, using D-alanine, and all these completely different methods, right, led to the same conclusion that one can see bacteria in tumors. But, you know, can I swear that every spot that we see is a bacteria? I probably can't, but that's what we see.
Yeah, it seems like that seems the challenging part of these experiments, right? Because of the possibility of contamination always. Okay, so I'm going to leave a few of these questions and we'll come back to them right at the end, if that's okay, Dr. Straussman, and then I will ask Dr. Ze'ev to come in and introduce our next speaker.
All right, thanks, Vasti. Thanks, Ravid, wonderful presentation. Tom, apologies ahead of time because I'm going to take your 50-page CV and I'm going to abbreviate it. And just briefly, Tom got his education at the University of Chicago, bachelor, PhD, MD, did PhD there as well, and then postdoc in Belgium with Thierry Boone. Came back to the University of Chicago where he has been leading melanoma oncology program. He's past president, Society of Immunology, Immunotherapy of Cancer, distinguished professor currently at the University of Chicago, and Abri Professor of Cancer Immunotherapy. Tom has been interested in studying anti-tumor immunity, understanding the mechanism of anti-tumor immunity, and translating his finding in the lab to clinical work. And that's one thing that very few among us can speak for. So Tom, thanks for being with us. It is all yours.
So thanks so much, Ze'ev, for the introduction and the opportunity to present here. So I want to build on a little bit what Ravid touched on in his talk, which is the ability of the gut microbiome to modulate systemic immune responses against cancer and thereby regulate cancer immunotherapy efficacy. And it might seem like a strange consideration, so I need to build the background a little bit. So the history of this is that we had been studying now for around 15 years the immunobiology of the tumor microenvironment and had identified that a large subset of human cancers has at baseline evidence for an ongoing smoldering immune response against the tumor that we've called the T-cell inflamed tumor microenvironment. These tumors are characterized by an array of chemokines, some of which are responsible for recruiting different immune subsets into the tumor, variable numbers of CD8 T cells, many of which we know are specific for tumor antigens, but many of those are not specific for tumor antigens. and are either passengers or recognizing other elements. And then a type 1 interferon signature that's connected to the innate immune pathways that are likely activated in order to generate that spontaneous adaptive immune response against the tumor. And at the time, this observation generated a philosophical question, why these tumors existed at all if they had all this good immune stuff going on. And it became clear that these are the tumors that have operational inhibitory pathways that turn those T cells back off. This includes the PD-1-PD-L1 interaction that everyone is now familiar with. And the non-T cell inflamed tumors on the other end of the spectrum lack any of these factors, and we have data based on specific mechanisms that many of these tumors actively prevent immune cell infiltration by different mechanisms. So as you might imagine, immunotherapy responders are enriched in this subset of tumors that allow activated T cells in and to do their work once negative regulatory mechanisms are interfered with. This is what these kinds of data can look by TCGA. These are about 160 transcripts that are all immune-related transcripts that are not specific to the tumor, but rather the host immune response. And in metastatic melanoma, about a third of these tumors have a rich set of immune regulatory genes. On the other end of the spectrum, it's about a third that are negative. And in between, there's a continuum, a little bit of a gradient. Many people call these hot, medium, and cold, or T cell inflamed, non-T cell inflamed, and intermediate. But suffice it to say, there's a spectrum, and it appears that immunotherapy response is enriched in the left side of this. So by immunohistochemistry, you can confirm some of these findings. The T cell inflamed tumors have CD8 T cells, but they're also the tumors that have the most abundant FOXP3 regulatory T cells that suppress T cell effector function. PD-L1 and IDO. IDO is another immune regulatory molecule that is involved in tryptophan degradation. And you can see the non-T cell inflamed tumors generally lack these factors. There are exceptions, but the idea is that this is an immune intrinsic negative feedback loop. The T cells come in, they produce chemokines that recruit Tregs. They produce gamma interferon that upregulates PD-L1 and IDO. The tumor cell isn't to be blamed for the negative regulation, at least these negative regulatory pathways. And so because of PD-L1 expression, you might predict that the T cell inflamed patients are more likely to respond clinically to PD-1 blockade, and that's indeed what's been observed. This is with the Merck pembrolizumab drug in a multi-tumor, multi-center setting, now taking a subset of immune genes, and you can see that this is now beyond melanoma. This is head and neck cancer and gastric cancer. Patients who have at baseline this activated T cell gene signature are the ones that are enriched for responders. This is progression-free survival on the vertical axis across these tumor types. And so this kind of observation begs the question, what are the mechanisms that explain the presence or absence, or let's say the tendency towards a T cell inflamed tumor microenvironment? Because if we could understand those mechanisms, then perhaps we could expand immunotherapy efficacy by modulating this process. And so what our group had done then, embarked upon a number of years ago, is to think about this kind of like a pharmacogenomics question. What are mechanisms of interpatient heterogeneity that could, theoretically contribute to whether a patient has a tumor full of CD8 T cells or not? And we had thought that this could, on the one hand, be represented by somatic differences at the level of the tumor cells. So oncogenic events that are on and off that could contribute to carcinogenesis, but also could contribute to immune regulation, either positively or negatively. Environmental differences and germline genetic host differences. We have evidence that all three of these are likely relevant. And I'm going to focus on the microbiome effect, but I do want to emphasize that in mouse preclinical experiments, in parallel with this work, we focused on what are the key mechanisms that drive a T cell response spontaneously against cancer. And here we found some key elements that a specialized subset of dendritic cells driven by the transcription factor BATF3, was necessary for cross-presentation of antigen to CD8 T cells. That upstream from BATF3-DCs, type 1 interferon signaling was necessary. And upstream from type 1 interferons was a dominant role of the STING pathway in host cells, largely in host antigen-presenting cells. And that the natural generation of anti-tumor T cell response in mice, at least, depended on STING pathway activation, type 1 interferons, and BATF3 dendritic cells that in turn led to T cell activation and tumor control that rendered tumors and the mice amenable to checkpoint blockade immunotherapy. Within the tumor cells, then, we found the first example was the Wnt beta-catenin pathway that could prevent these three processes from happening. We now have germline genetic differences I won't have time to talk about that also contribute to regulation of some of these processes. And what I'm going to focus on today is the microbiome. So this is the reason we started focusing on microbiome as a variable, because there's tremendous intrapatient heterogeneity, or between mouse colonies, inter-mouse colony heterogeneity that could theoretically contribute to variation of the natural immune response against the tumor and the degree of this immune infiltration that's seen. And so why did we think that the microbiome could have been a candidate source of heterogeneity in the host? And that's because we view spontaneous anti-tumor immunity a little bit like autoimmunity. And it had been shown in a series of publications by Dan Littman and others that the composition of the gut microbiome could impact more globally on immune responses in the host, not just mucosal immunity in the gut, but in distant sites as far away as in the brain or in the joints in a rheumatoid arthritis model, for example. And so we wondered then whether in mice the composition of the gut microbiota can contribute to anti-tumor immunity. And we borrowed as a model system one which Dan Littman had utilized, which is mice purchased from two different vendors that he had already characterized as having distinct microbial communities, and basically bought mice from these vendors and plugged them into tumor models. And these data were published several years ago, so I'm not going to dwell on them too much. But these mouse vendors were Taconic and Jackson Labs. And Taconic mice, we always found a reproducible, faster tumor growth compared to Jackson mice. And this is a modest difference, but it was consistent. And this was associated with about a 2 or 3-fold increase in the endogenous CD8 positive T cell response against a tumor or defined tumor antigen in that system. This was microbiome related. I don't have the data here, but we showed that you could eliminate these tumor growth differences by co-housing of mice or by fecal transfer back and forth. And what we found was that fecal transfer from Jackson mice into Taconic mice could improve tumor control and the anti-tumor immune response. So it was a gain of function in Jackson mice in this case. So that was exciting to us because it generated the possibility that maybe there could be a therapeutic approach to improve anti-tumor immune responses and immunotherapy efficacy. And in this series, we identified about a dozen bacterial entities that were enriched in Jackson mice that upon transfer into Taconic mice, one of them was Bifidobacterium, and we identified a bit of Bifidobacterium preparation that could partially control tumor growth in the Taconic mice, which are the ones that lacked favorable gut microbes. This therapeutic effect in established tumors, by the way, was on par with what we saw with anti-PD-L1 antibody. Now, when we put them together, we improved efficacy up to the point, sort of a condition that we saw naturally in Jackson mice that had a favorable microbiome. And in this case, the entire benefit was immune related, not driven by a direct anti-tumor effect, because we lost that when T cells were depleted. And we worked out some of the mechanism here that was linked to better T cell activation with Bifidobacterium in this case. So Ravid alluded to the human translation of that work. Then we decided to add to our human metastatic melanoma biobanking activities stool sequencing. And here we did, this is 16S sequencing, but we did shotgun sequencing and then used confirmatory species-specific PCR for about 10 of these bacteria. And we found indeed that there were bacterial sequences enriched in responders to anti-PD-1. This is at baseline before they got treatment. And this is stool 16S sequencing, not the tumor. But there were also other bacterial sequences enriched in non-responders. And now we believe that both of these are functionally relevant, that there might be favorable bacteria. We have functional evidence for favorable bacteria, but also for unfavorable bacteria that at some level might interfere with this process of anti-tumor immunity and anti-PD-1 efficacy. How do we know that those bacterial compositions are causal for improved efficacy? Well, it can't be the only mechanism, because we know that there are tumor cell intrinsic and host genetic factors that also contribute. But in many cases, it turns out in about 50 to 70% of the cases, we can line up anti-PD-1 efficacy by transfer of fecal material back into germ-free mice that lack their own microbiome. But it doesn't. Is not recapitulated in all cases, but it is in many cases. And this is one of these examples. Patient B was a non-responder to anti-PD-1. When we transferred that patient's fecal material into germ-free mice, let equilibrate for a couple of weeks, and then put in a mouse tumor and treated with mouse anti-PD-L1, the mice were non-responsive. And conversely, patient A was clinically a responder. Transfer of fecal material from that patient into germ-free mice already was slowing tumor growth to some extent, and then those mice then became responders to anti-PD-L1. And so these data were quite exciting for us. It argued that in many cases, the composition of the gut microbiome is rate limiting and might be a dominant effect. And the fecal transfer data argue that perhaps we could intervene and change the natural biology and improve therapeutic outcome. And there were two additional papers from Jen Margot's group at MD Anderson and Laurent Sidvogel's group in Paris with similar patterns, although the specific bacteria are different. And this is a concept to come back to perhaps in the discussion. So, so we wanted to build on this observation or these observations. And so the next questions were basically in two categories. What are the exact bacteria that are either good or bad in patients that might be manipulatable towards therapeutic efficacy? And then what's the mechanism? If you think about it, we're considering how bacteria in the gut can impact in a long distance way an immune response on the other end of the host in metastatic sites. And so how might this long distance immune regulatory effect occur? Ravid was talking about intratumoral bacteria, and I'll tell you in our models with these two conditions, our Taconic versus Jackson model and in the reconstitution of germ-free mouse model, we do not identify the gut bacteria within the limits of detection within the tumor in these experiments. And Ravid also mentioned, in short-term transplantable tumor experiments, you might not see that. These are, experiments that take only about 3 weeks overall. And so we're not saying intratumoral bacteria are not important, but they're unlikely to explain this mechanism of potentiation by gut bacteria at the immune level. So we wanted a model system where we could do longer-term experiments to do reproducible experiments, replicates, not these single one-off germ-free mouse reconstitution. So what we decided to do was try to reconstitute these germ-free mice acutely as adults, but then breed the mice among separate colonies and see if the phenotype of anti-PD-1 response or non-response could be maintained and passed longitudinally And if they could, then we could do multiple experiments, larger ends in our experiments, and then that would give us a more stable model for determining mechanism. And we're concerned about this because, first of all, germ-free mice from the get-go have immune developmental defects, and it's been reported that they can have a leaky gut barrier. And so putting bacteria directly into adult germ-free mice might not be the same as naturally acquiring bacteria from birth from the parental generation. So pictorially, this is what we're doing. We make these avatar mice, but then we're breeding, the A mice and the B mice together to see if we can transfer the phenotype. And in some cases, this is quite evident, where you can pass the phenotype generationally. So these are the originally colonized mice, the first generation, the second generation, and you can see that this looks like it's passed by generation. It looks like a genetic trait, where these mice, the only difference is they were born from parents that were reconstituted with microbiota from a non-responder melanoma patient versus a responder melanoma patient. So this phenotype can be propagated generationally, and it looks like a genetic trait, but it's actually an environmental trait, and this gave us a stable model system. So we've approached these questions then in a variety of ways. We're doing culture and sort of bacteria function phenotype experiments to try and identify the bacteria. And one approach has been to narrow the field of those bacteria is to study cross FMT between these mice. I'll say on the onset that along with stability of the phenotype, when you do 16S sequencing on these generations of mice, the composition is also stable. Not every bacteria from the original patient donor is stably reconstituted. That's been well described in literature, but the phenotype is maintained. So we think that the functionally most important bacteria are maintained in order to recapitulate the responder, non-responder phenotype. So what happens if we do an FMT back and forth? Can we correct the defect in B mice? And the answer is yes. This is the normal progressive tumor growth we see in these B mice. And now and this is what we normally see in A, if we give 2 courses of fecal transfer from the responder mice, we can now slow tumor growth and improve. All these mice get anti-PD-L1 and improve anti-PD-L1 efficacy. So this argues it is possible to correct a microbiome defect even when it's inherited, such as this, and improve therapeutic efficacy. But what about the other way around? This is the normal tumor control we get in A mice. But if we do FMT from the non-responder mice, we can also screw this up. We can lose the therapeutic efficacy, meaning that you can disrupt, at least in this example, the favorable gut microbiome by transfer of an unfavorable microbiome. And FMT is not always a positive effect. And we need to keep this in mind with our clinical translation of FMT. We have to be careful who the recipient is, and we likely have to be careful which donor we choose so we don't mess it up. But these data also argued that there's a little bit of a yin-yang going on. You can have kind of everything tipping in your favor, but then if you inappropriately manipulate the microbiome, you can disrupt this favorable immune response. So our mechanism in this case has to account for kind of a yin-yang seesaw phenomenon. So the way we decided to approach this then was starting from the tumor side. So I have a student working from the tumor microenvironment side and a postdoc working from the gut bacteria side. And we did this by doing single-cell RNA-seq of all the immune cells in the tumor microenvironment. And you get roughly about 30 subsets of CD45 positive cells in the tumor microenvironment. When you compare them overall between the A mice and the B mice, there's less than a 2-fold difference in the quantity, the percentage of each of these subsets. But what changes quite dramatically is the gene expression profiling of specific subsets. One of them that you might expect is the gene expression signature of CD8 positive T cells themselves. We know that the CD8 T cell response is accentuated with favorable microbiota. And as you might expect, the responder mice, the favorable gut microbiota mice, their CD8 T cells have increased expression of different activation markers, including PD-1, LAG3, and TIM3, which are purported negative regulators, but also positive regulators. And I've thrown one of these in here, 4-1BB. And we know from previous experiments in transplantable tumors in mice, these are markers of tumor antigen-specific T cells that are trying to be activated against tumor antigens. And these data argue that this phenotype is pushed by supportive microbiota. But what's more striking is looking at myeloid cells. And I'm just going to show a couple of examples here. When we look at the macrophage subsets, which fell into these clusters, what was evident was that the good microbiome mice have an enrichment for an M1 phenotype of macrophage, whereas the quote-unquote bad microbiome mice have an enrichment for an M2 phenotype. And this could be confirmed by flow cytometry. We pulled out just one marker in each dimension. CD206 as an M2 marker. You can see we have more CD206 positive myeloid cells. These are gated on class 2 high F480 macrophages in B mice, and conversely more CD86 positive macrophages in A mice. And an M1 to M2 ratio is quite powerful at sort of predicting the A versus B microbiome phenotype. The other dimension, the other cell subset that showed a remarkable shift in gene expression pattern were the granulocytes. And the granulocytes from B mice had expression of what has been published as an MDSC-like granulocyte population. In contrast, the granulocytes from A mice, the favorable microbiome mice, have more of a conventional neutrophil gene expression signature. And one marker that is probably hard to see, but where the arrow is CD44 had recently been published as a surface marker that's higher on MDSC-like granulocytes. And by flow cytometry, that's one way we could confirm in independent experiments enrichment of this MDSC-like neutrophil population. So we think the B mice have the unfavorable microbiome mice have a higher suppressive myeloid population, and the favorable microbiome mice have more of a anti-tumor type 1 T cell activated macrophage signature. So what Jessica did then was go back to a human cohort, and this is just a subset of our melanoma patients treated with anti-PD-1 where we have their tumors at baseline. And she looked at a gene signature indicative of M1 and M2 ratio. And what you can see is that the responding patients have a higher M1 to M2 ratio gene signature compared to the progressive disease patients. And this you could quantify even in a different way. If you look at the change in tumor size, so tumor shrinkage versus growth with treatment, these are actual patients. The progressive disease patients with growth of their tumor, these have a higher M2 to M1 ratio, and the responders have a trend towards a higher M1 to M2 ratio. So this is one mechanism that we have landed upon. We've confirmed this in a second mouse tumor model, and we're working with other patient-derived samples to see if that's confirmed in individual patients. And this is a mechanism that makes a lot of sense to us. Bacterial-derived products are known to impact on myeloid cells, both granulocytes and macrophages. We know TLRs affect macrophages and so on. And so this is a mechanism that we're quite attracted to that could be broadly applicable and also could explain some of the details with our clinical translation, suggesting that it's not just one bacteria type that's important, but collections of bacteria could have positive or negative effects. So we're working on the mechanism, moving upstream. one likely candidate is metabolites that might be derived from those bacteria. And now, by having reconstituted mice where we have at least 5 mice per group and we can do replicate experiments, we can now do serum unbiased mass spec to start to identify metabolites enriched in the responder mice or enriched in the non-responder mice. And then by doing the FMT back and forth, look for the subset of metabolites that are perhaps being derived from the bacteria that have either positive or negative impact on myeloid cells that in turn regulate the immune response at the level of the tumor microenvironment. And by integrating these different dimensions, the metabolites, the shotgun sequencing of the bacteria to look for pathways, the microbiome composition of these reconstituted mice, and the myeloid cell gene expression signature, we're narrowing in on the mechanism, which now, just to remind you, this is just one patient pair, A versus B, and we have multiple of these patients who could have additional mechanisms that go beyond the myeloid gene signatures. And then based on, finally, Ravid alluded to this, but I do want to mention that there are multiple clinical interventions being pursued to try and, let's say, correct an unfavorable gut microbiome in patients towards an immune potentiating one. This includes specific bacterial preparations, like the bifidobacterium preparation developed by Evelo, and FMT from responding patients into non-responding patients, where also, these are the first papers, this is the Israeli paper showing 3 clinical responders, along with favorable immune changes in the tumor microenvironment in anti-PD-1 failures, arguing that in some cases the microbiome can be manipulated favorably. So I tried to argue for the importance of the tumor microenvironment, the different sources of interpatient heterogeneity, the gut microbiome being one of those differences, and how maybe we can use this as therapeutic intervention. And I'll just end with the acknowledgements here. This is the group focusing on the microbiome component that I outlined today. So thanks very much for your attention, and I guess there's time for questions.
Yeah, let's do a few questions because we're getting quite a few coming in. So firstly, and I think this is a really interesting question, is in the mouse models, it looks like when you have responder versus non-responder microbiota, it affects just the baseline growth of the tumor, even without therapies. Is this true in patients, that you could basically use it as a prognostic marker based on their gut microbiota?
Not clear. So in our in these transplantable tumor models, in our experiments, we do see a slight improvement in tumor control. That's all immune dependent, and so you know, if we get rid of the host T cells, you know, those curves are squashed, and so you know, in the specific examples where the patient's immune response is better against their tumor, we already know that that has positive prognostic import for cancer, especially melanoma. So that correlation is there in patients. If patients lack an immune infiltrate, they do worse just prognostically. And that's true in early stage melanoma, true too without any treatment. But then additionally, you add immunotherapy, and the responders are enriched in the patients who have some kind of ongoing immune response.
Yeah, that makes sense. Actually, Dr. Straussman, can I get you to turn on your camera and unmute yourself as well? Because there's actually an interesting pair of questions for both of you that I think we could get your insight on. So is it possible to predict the intratumoral bacterial composition based on analyzing the gut microbiota of the patient? I honestly don't know which one of you can answer better, but maybe you both together can give us the best answer.
We don't have good enough data for that. For the first time, we have now a cohort in which we have both the gut and tumor microbiome of patients, but we haven't analyzed it enough. So I do not know the answer.
Yeah, same here. I mentioned that in our mouse models, in these short-term, you know, 3-week experiments, we don't see evidence of gut bacteria in the tumors. But we now have, you know, total shotgun sequencing of all these tumors from those patients, from most of the patients, N of about 60. And we know we can detect bacterial sequences in some of them. So that's a question for the future, to try and decipher this. Now, my prediction, right, is that if it's true the tissue of origin is contributing to the bacteria that are inherited by the tumor as it metastasizes, my guess is in melanoma, which start these cutaneous melanomas start in the skin, we'll probably see bacteria that look more like skin-derived bacteria in the metastases that originated in the skin, and that the biology of intratumoral bacteria might be distinct from the biology of gut bacteria. Gut bacteria, just by sheer mass in the host, is more likely to regulate systemic immune responses, right? It's just the volume, the mass effect of those bacteria, plus the clear connection between gut mucosal immunity and systemic immunity, argue that the systemic anti-tumor immunity is most likely regulated by gut bacteria, or at least, let's say, dominantly affected by gut bacteria. But my bet in the lab is that any tumor-associated bacteria we see in metastatic melanoma might be skin bacteria. Maybe Ravid has data already, but I'll see if I win my nickel bet on that. Yeah.
The only thing I can add is that we have a small cohort of patients, of melanoma patients, for which we took more than biopsy for more than one tumor. And indeed, in these different metastases, we see the same bacteria, or very similar bacteria in these tumors. So they should probably originate from the primary tumor and traveled with the tumors. Also a paper, a really nice paper, demonstrating that colorectal bacteria sometimes travel with the cancer cells to the metastatic site. So yeah, it makes a lot of sense.
Complicated. Actually, I have a question. I'm really sorry for all the questions we're not able to answer in the interest of time. But you both sort of alluded to this idea that potentially the genetic changes, whether germline or somatic, in the tumor might have an effect on the microbiome. And Dr. Gajewski, what do you think of, like, you know, these familial cancers where you have like Lynch syndrome or these sorts of things where you have a germline variant in the person, which gives them a predisposition, and we know that it gives them a predisposition to certain cancer types. Do you think that those sorts of germline variants could actually affect the gut microbiota, or is that just too fanciful?
It's a theoretical possibility. I mean, part of our overall, like the mad scientist grandmaster biomarker plan is to integrate the germline DNA data, the tumor sequencing data, the microbiome data, the serum metabolite data, responder, non-responder, anti-PD-1, and see what's dominant in individual patients. So this is what we're working on. It's taking longer than I'd hoped, and part of that is a bioinformatic challenge. But also, we don't have all of the elements defined appropriately yet. You know, what are the features that we need to sort based on? But that being said, so we don't know the answer. There's one paper that I know of, which is from the Weizmann Institute, about, you know, in the diabetes context, where diet-specific interventions have variable impacts on blood sugar based on microbiome composition and linked to the individual patient. There, Ravid can correct me if I'm wrong, but the working model or the evidence was that germline variation had a minimal effect connected to the microbiome composition. That's my recollection of those data. And so it's theoretically possible that there could be germline variation that can impact on microbiome, at least in these kinds of cohorts that hasn't been seen. I want to propose an exception, a likely exception to that, though. There are known immune regulatory genes that are linked to mucosal immunity, right? That are connected to inflammatory bowel disease, for example. And one might predict that some of those might, in a circuit, change microbiome composition because of mucosal immunity against microbes, or some microbes that might change the balance of the microbial community in the gut, maybe bacterial peptides or what have you, that might change the composition, or specific immunity against some bacteria that then change the composition. So I think it's theoretically possible in those cases where there's an altered mucosal immune response that's impacted by host genetics. So I would propose that as a reasonable hypothesis, but that in most cases, I think that effect's probably going to be minimal, and there'll be independent traits, right? Well, we know that we have a couple germline variants that are clearly connected to efficacy. I doubt that efficacy is via the microbiome, although it's formally possible.
And in parallel, Dr. Straussman, what would you say for the intratumoral microbiome? Do you think that could be affected by certain somatic events or germline events during tumor progression?
Yeah. So I would say that we have some unpublished data in which we look at specific tumors like ovarian and breast cancer. We compare BRCA mutated and as a germline. And we do see some differences in the microbiome of these tumors. But these have, so that might be an example in which germline mutation affect the composition of the tumor microbiome. There were also nice papers demonstrating, for example, the p53 status of tumors, specifically in the lung, can affect the composition of the microbiome. I think this was somatic mutations, if I remember correctly, rather than germline mutations. But I can see how the composition, the genetic composition in the tumor can affect, the microenvironment of the tumor and select for different bacteria. So maybe it's easier to see this happening in the tumor rather than in the gut.
All right. In the interest of time, we're probably going to have to stop here. Again, apologies for any questions we couldn't answer. And thank you, all of the attendees, for staying late. But I know I don't really have to thank you because it was a fascinating set of talks and discussion. Just as a reminder, March 8th is the next session. So please tune in, register. I think it's also going to be fantastic. Spread the word, social media. This is going to be, I think, a really interesting set of talks for the next 6 to 9 months.